Morning, all of you. Uh, myself, Dr. Srinu Balla. So today we are going to discuss about gene cloning strategies. So what gene cloning strategy means? The gene cloning strategy means simply a set of techniques adopted for gene cloning for a particular purpose is said to be a gene cloning strategy. సో మెయిన్ ఈ జీన్ క్లోనింగ్ స్ట్రాటజీ అంటే ఏంటి అంటే సర్టెన్ పర్పస్. సో ఇఫ్ యూ డూ ద జీన్ క్లోనింగ్, సో ఆ జీన్ క్లోనింగ్ చేయడానికి యూస్ చేసే టెక్నిక్స్ మనం అడాప్ట్ చేసుకొని సర్టెన్ పర్పస్ అంటే ఇక్కడ మనము పర్టికులర్ జీనోమిక్ లైబ్రరీ ప్రిపేర్ చేయడానికి ఏదైతే స్ట్రాటజీ అనేది యూస్ చేస్తామో దాన్ని జీన్ క్లోనింగ్ స్ట్రాటజీ అంటారు. Coming to the what mean by genomic DNA library? A collection of clones containing all DNA segments of the genome of an organism is called a genomic DNA library. So another example, so the genomic library is created by inserting all fragments of a genomic DNA into host cells using vectors. So in the gene cloning, the main method main method, shotgun method. So this is shotgun cloning method is a cloning strategy which has been done for constructing genomic DNA library. So what mean by gene short cloning technique or shotgun cloning technique? So cloning all the DNA fragments of a genome into cleaved vector at a time by a single cloning experiment is called shotgun cloning. So ఈ shotgun cloning technique లో all the DNA fragments is cleaved and introduced into the vector, and after that a single clone by a single clone, the experiment is called shotgun cloning. So there is two approaches are made on the shotgun sequencing. One is whole genome shotgun sequencing. Second one, hierarchical shotgun sequencing. So uh, I want to explain in detail, briefly about this shotgun sequencing. In this shotgun sequencing, in this shotgun sequencing, సో ఇప్పుడు మీకు ఏది ఎక్స్ప్లెయిన్ చేస్తున్నానంటే షార్ట్గన్ సీక్వెన్సింగ్ ఎలా చేస్తారు అంటే, సో హియర్ ఫస్ట్ మనము ఏం చేస్తాము అంటే, సో వీ హ్యావ్ టు టేక్ ద ఆర్ వీ హ్యావ్ టు ఐసోలేట్ ద జీనోమిక్ డిఎన్ఏ. సో జీనోమిక్ డిఎన్ఏ అనేది ఫస్ట్ వేరియస్ మెథడ్స్ యూస్ చేసి జీనోమిక్ డిఎన్ఏని మనము ఐసోలేట్ చేస్తాం. ఇలా ఐసోలేట్ చేసిన జీనోమిక్ డిఎన్ఏని by using restriction enzymes, by using various restriction enzymes, we will be cleaved into the fragments. So we are cleaved into the fragments restriction enzymes యూస్ చేసి ఈ జీనోమిక్ డిఎన్ఏని మనం ఏం చేస్తాము అంటే ఫ్రాగ్మెంట్స్ కింద డైజెస్ట్ చేస్తాం. దెన్ దీస్ DNA fragments are separated by using electrophoresis. So here we have to use electrophoresis technique to separate these DNA molecules based on the size. So mass by charge ratio, these DNA fragments are separated on the gel based on mass by charge ratio. So on the gel, the DNAs are separated based on the size. Later on, these DNAs, so which DNAs, if you want to clone the, if you want to clone the specific size of the DNA, so which size of DNA, or if you want to clone that, so to be cloned DNA, we have to choose it. To be cloned DNA, we have to choose it. సో ఇలా చూస్ చేసుకున్న తర్వాత దెన్ అందులో లార్జర్ ఫ్రాగ్మెంట్స్ కానీ స్మాలర్ ఫ్రాగ్మెంట్స్ కానీ మనం ఏం చేస్తాము అంటే డిస్కార్డ్ చేస్తాం. సో ఏవైతే రిలయబుల్, ఏదైతే రిక్వైర్మెంట్ ఉన్నాయో ఆ డిఎన్ఏస్ని మనం ఏం చేస్తాము అంటే సెలెక్ట్ చేసుకుంటాం. సో ఇక్కడ తీసుకున్న డిఎన్ఏస్ ఏవైతే ఉన్నాయో ఈ డిఎన్ఏస్, okay, these DNAs are treated with alkali, okay. So alkaline phosphatase enzyme, alkaline phosphatase enzyme we have to take. So this alkaline phosphatase enzyme remove the 5 prime phosphate at the this DNA. So remaining it has a at the 5 prime side 5 prime OH, 5 prime OH. So 5 prime OH only present at the 5 prime side, and 3 prime side obviously it has a 3 prime hydroxyl group. Now, so we have to choose the vector. So this is vector. So this vector we have to uh, restricted by restriction enzymes. సో ఇక్కడ ఏ restriction enzymes అయితే యూస్ చేసామో for the genomic DNA restriction side, here also same restriction enzyme used for the vector restriction. So we got restricted DNA, restricted DNA. So this, after the restriction of this DNA, we have to ligate these two DNAs. So here vector DNA, here genomic DNA, we have to ligate. After ligation, after ligation, the vector DNA with the vector DNA, the genomic DNA is ligated, it will form the rDNA, recombinant DNA. So this recombinant DNA we have to transformed into the E. coli. So we have to transform into the E. coli. We have to transform into E. coli. E. coli లోకి ట్రాన్స్ఫర్ చేస్తాం. సో ట్రాన్స్ఫర్ చేసిన తర్వాత then we have to choose whether our recombinant DNA consists E. coli cells. So for that we have to screen the cells. So we have to screen the cells. After screening, so which E. coli cells consist the recombinant DNA, so we have to isolate it. We have to isolate it, and we have to produce. We have to place it on the uh, media, and it will produce the number of copies of our genomic DNA. So after that, we have to isolate these genomic DNA, this genomic DNA, this genomic DNA, and we have to done the chromosome walking process for the sequencing of DNA, chromosome walking. So this is the process of genomic DNA. So genomic DNA libraries we can be prepared like this. So coming to the, so coming to the shotgun cloning method, the following steps are its first, genomic DNA is isolated, genomic DNA is isolated from the source of organism. Isolated genomic DNA is cut with the restriction enzymes to generate small DNA fragments. The restriction digest is electrophosed on a polyacrylamide gel in the presence of molecular weight markers in one lane. DNA fragments of clonable size are isolated from the gel and the smaller as well as larger fragments are discarded. For example, if the DNA fragments have to be cloned in plasmid, size of the fragments should be between 5 and 15 kb. So fragments lesser than 5 kb and larger than 15 kb are discarded. So after that, the isolated DNA fragments are treated with the alkaline phosphatase to remove 5' phosphate groups from the ends of the DNAs. The vector DNA is cleaved with the same restriction enzyme which is used to digest the genomic DNA. The dephosphorylated DNA fragments are mixed with the linearized vector DNA to construct rDNAs. The rDNAs thus constructed are introduced into the E. coli or yeast. The recombinant cells are separated from one another using dilution plating and subsequent culture. So, the microbial cultures are screened to detect the contigs. Based on the contig analysis, the clones are arranged in the genomic library. So here the advantage is much more economical and faster, labor saving because the sequencing reaction is virtually fully automated and the sequences being assembled by the computer programs. So disadvantages put a large requirement on the ability to align the overlapped fragments and reconstruct the chromosome complex process. So thank you.